When feeding on infected hosts, female sandflies may ingest mastigotes with the blood meal, some of which may divide and transform into promastigotes. Here, a mammalian macrophage infected with mastigotes is being drawn through the sandfly pharynx, esophagus, and thoracic midgut or cardia, and into the gut by synchronized sidereal and stomatidal pumps and valves. Typically, pumps are synchronized so that while the sidereal valve is open and stomatidal valve is closed, the suction creates unilateral flow. Once the stomatidal valve opens and sidereal valve closes, this unilateral flow continues onto the gut. The extrinsic cycle of the Leishmaniasis parasites is divided into 3 stages. It is 6 a.m., 12 hours since the last blood meal. In the first stage, 0 to 2 days post-feeding, all parasite development occurs in the blood meal within the parasitrophic membrane in the midgut. During a mastigote to promastigote transformation, research suggests that mammalian blood meal may contain a transformation blocking factor that is gradually digested by phlebotomous enzymes. There will be a minimum of 2 divisions between the 0 to 2 day transformation time period, while mastigotes develop to more motile procyclic promastigotes. Procyclids are short, ovoid, slightly motile forms with large diffuse nuclei and short flagella, typically seen swimming about the periphery of the blood meal. Some may be seen dividing by binary fission. Binary fission is a form of asexual reproduction and cell division not involving the formation of spindles, where single DNA molecules first replicate, then attach each copy to a different part of the cell membrane, which is then followed by original chromosome replication, consequently leading to 2 cells being genetically equal. Each division evolves a parasite, mass producing and specializing it for a specific purpose. Between 36 and 48 hours, these early promastigotes begin to transform into long, slender, highly motile forms with long flagella. After the first stage, and at the beginning of the second stage, 2 to 5 days post-feeding, the parasitrophic membrane ruptures and the partially digested gut contents begin their passage through the pylorus and into the ileum. This event is accompanied by an intense multiplication of procyclic promastigotes and transformation into nectomonads, which fill the anterior abdominal midgut. The parasitrophic membrane consists of a chitin framework and a protein-carbohydrate matrix. Strong evidence supports that this membrane is mediated by chitinases secreted by the Leishmania promastigotes. Consequently, the parasitrophic membrane ruptures and allows an eventual forward migration of the parasites to the phlebotomous mouthparts. Many nectomonads attach themselves by interdigitating their flagella with the microvillar lining of the gut wall, and in this way avoid being passed out with the digested blood meal. Research has determined that midgut attachment of promastigotes is controlled by the expression of terminally exposed galactose residues on lipophosphoglycan, or LPG, the major promastigote surface molecule. There is also evidence that gut lectins, or lectin-like molecules, serve as parasite attachment sites, and the stage-specific expression of complementary sugars on LPG provides an underlying mechanism controlling the retention of attached, dividing parasites during blood meal digestion and excretion. This expression of sugars also would control the release of infective stage permastigotes, permitting their anterior movement so as to be positioned for transmission by bite. As the blood meal diminishes, parasites migrate forward into the thoracic midgut and transform into actively dividing haptomonads and paramastigotes. Haptomonads and perimastigotes attach themselves to the cuticular intima of the cardia and stomadia valve by foot-like enlargements of their flagella called hemidesmosomes, or they bond to each other by a gel-like matrix. Utilizing desmopenetrant cell adhesion proteins, hemidesmosomes, or HD, are comprised of two rivet-like inner and outer plaques together with the anchoring fibrils and anchoring filaments, which are collectively termed the HD stable adhesion complex or HD anchoring filament complex. During the third and final stage, 5+ days post-feeding, the gut is cleared of the blood meal, egg development is completed, and oviposition begins. Concomitantly, a massive infection of dividing haptomonads, paramastigotes, and nectomonads erupts at the stomatal valve, forming a darkened lens or ball-shaped mass, effectively impeding the flow of blood into the midgut. Research has observed deterioration of the cuticular lining of the stomatal valve of various species, presumably caused by chitinolytic enzymes secreted by parasites attached en masse to the valve. While the sidereal and stomatal pumps expand in unison, the sidereal valve has to be open and the stomatal valve closed to permit unilateral flow. A damaged stomatal valve from chitinolytic enzymes remains open, and therefore the pump sucks in both directions, causing regurgitation of midgut contents mixed with parasites. This is a clearer view of the interior of the ball-shaped mass. Note the dividing haptomonads and paramastigotes. Many of the haptomonads and paramastigotes will migrate forward into the esophagus and posterior pharynx. Note the infective migration of the haptomonads and paramastigotes into the esophagus and posterior pharynx. Metacyclogenesis has been described as a pre-adaptation to survival within the vertebrate host. Unlike other developmental forms which are immobilized because of attachment to or entrapment around the stomatal valve, metacyclic permastigotes are unattached and highly motile and are able to migrate anteriorly past the stomatal valve into the pharynx, cibarium, and proboscis.